Okay, good afternoon, everyone. My name's Rachel Syme, and I'm the Vice Chair of N2, the Network of Networks, and it's my great pleasure to welcome everyone to today's N2 webinar. This is the second in what we're calling our COVID series. And as this webinar has been opened up to more than just the N2 audience or the N2 members, I thought I'd say a few words about N2 before we turn it over to today's speaker. So N2 is a not-for-profit organization, a real alliance of Canadian research networks and organizations, really working to enhance Canada's clinical research capability and capacity. N2 provides a lot of platforms for sharing best practices, education resources, and research-related content, as well as acting as a real national voice and advocate on behalf of a really wide range of clinical research and clinical trial stakeholders. And currently there's about 150 member organizations in N2 from coast to coast. So with our focus as Canada's Alliance for Excellence in Clinical Research, it's really appropriate that the focus of today's talk is the importance of scientific rigor during crises. So with that, I would like to introduce our speaker today, Dr. Jonathan Kimmelman. Jonathan is a James McGill Professor in the Biomedical Ethics Unit, Social Sciences of Medicine, with a cross appointments in experimental medicine, epidemiology, biostatistics, and occupational health and human genetics. He holds a PhD in molecular biophysics and biochemistry from Yale University and joined McGill in 2005. His research revolves around the ethical, social, and policy dimensions of translational research. And I can say, I first met Jonathan at a CIHR New Investigator meeting in Mont-Gabriel, Quebec, where he was talking about the reproducibility of science. And that's where I learned what an engaging speaker he is and the interesting perspectives that he has to share. He recently co-published a short article in Science entitled Against Pandemic Research Exceptionalism. And we really felt at N2 this would be a great topic for a webinar. And we're very fortunate he's agreed to speak with us today. So before I turn things over to Jonathan, I just want to say if you do have questions, please use the Q&A function of Zoom, and we'll be addressing the questions that way. And with that, I'd like to turn the virtual podium over to Jonathan.
Great, thank you very much. I am going to try to, yep, here we go. Okay, can everyone see my slides? Or at least one person?
Yeah, you're good.
Okay, great. So thank you so much for the honor of this invitation. I thought it would be good to start off with some really basic background about human research ethics. My talk's going to divide into 5 parts, but I'm going to start with the rudiments, and then I'm going to work my way forwards to some specific issues we're encountering with clinical trials in COVID-19. So, okay, good. They're just having problems with controls here. Ah, good, we're good. Okay, great. So drugs, vaccines, devices, these are truly wonderful things. If you add up, for example, the number of years of life lived as a result of, for example, highly active antiretroviral treatment, at least a few years ago, it came out to about 2.8 million years accumulated. And that's largely due to the advances we've made in understanding how to treat HIV. Likewise, if you go to an area like pediatric cancer, survival rates in the 1960s were somewhere on the order of 10%. Today, 5-year survival rates are probably higher than 80% for children. And again, that's due to the way that we've been able to harness the power of science to improve therapeutic options for children. But drugs, devices, vaccines, they all have a dark side. One of those dark sides is that they all have side effects. They account for between the fourth and sixth leading cause of death within hospitalized patients, adverse events caused by them, that is. And as well, they're incredibly expensive. In the United States, about $404 billion last year were spent on prescription pharmaceuticals. They account for a sizable fraction of our healthcare expenditures. Every dollar we spend on healthcare is one less dollar we have to spend on all sorts of other things that we might care about in a society. And so, accordingly, when we want to roll out drugs, devices, vaccines, it's critical that we have good evidence to establish that those costs and risks are going to be redeemed by the prospect of benefit for using those. And the challenge there is that in order to derive that evidence, we have to do experiments, not just any kind of experiments, but we have to experiment with human beings. And that, as we all know, is an ethical minefield. And it's an ethical minefield essentially for two reasons. The first reason is that the very reagents that we use in that research have moral status. It's unlike a field like geology, where you can crack open rocks to your heart's delight and grind them up and put them in spectrophotometers or whatever you want to do with them, and they won't complain. When you do that with patients, you are potentially setting back their interests, you're causing pain potentially, and accordingly, we need to find ways to experiment with human beings that are going to respect those human beings. And so a lot of what we learn when we learn about human protections and research ethics is all about creating this membrane around the human subject that protects them from undue incursions from society and research in the name of scientific advance. Now, there's a second issue that's at stake when we do medical research, one that doesn't get as much billing as it should, but it's really just as important. And it's the issue that the information that we generate when we experiment on patients almost literally is ultimately ingested by downstream users of that evidence. We make all sorts of life and death decisions based on the results of clinical trials, much in the same way that we ought to care about the chain of custody as food goes from the farm all the way to the table. Similarly, in medical research, we have to really be concerned about protecting evidence as it goes from the clinic through publication to downstream users, protecting that evidence from adulteration, from bias, or any other sorts of outside interests that could distort our decision making. So a second component of research ethics really has to do with creating a sphere of protection, not just around the human volunteer, but also around the integrity of the evidence that we generate. And so what I want you to take away from this section is just simply the idea that when we do research ethics, the ethics and the science are intimately intertwined. We can't really partition human protections from the nature of the task of generating reliable evidence in front of us. Okay, so let's move on to part two. I want to talk a little bit about drug development and some of the challenges we have when we are trying to develop novel therapeutics. And that will eventually get me to talking about novel therapeutics around COVID-19. So probably everyone in this webinar knows that drugs typically progress through a series of clinical trials, phase 1, phase 2, phase 3, before they ultimately get licensure by a regulatory authority like Health Canada. And probably many people here know that most of the drugs that start off that process ultimately fail to vindicate the efficacy and safety that were observed in animal as well as small clinical trials. So only about 11% or 14%, depending on the estimate, of drugs that start off that journey from phase 1 ultimately lead to a regulatory approval. In some areas, the numbers are actually quite lower. Oncology, for example, it's about 5%. Now, what's also important to remember is that of those drugs that get regulatory approval, not all of them actually have major impact or benefit for patients. Estimates vary, but probably about a third to a little bit more than a half of drugs that get regulatory approval, at least in the United States, deliver medium or high value to patients. So a lot of drugs that get approval, they're marginal value. They're not necessarily dramatically changing the lives of patients. So the broad point that I want everyone to understand here is that it's incredibly hard to go from an idea, from a molecule, from preclinical research, not just to a regulatory approval, but to a regulatory approval of a drug that actually is transformative of our welfare and lives. And there are a number of different reasons why that's so, but some of them are just hard statistical and scientific realities. So I think one of the issues is that a lot of the research that we do when we do clinical trials is at the cutting edge of what we actually understand about the mechanism of disease. Let's take a disease like Alzheimer's, a drug like semagacestat, which was put into clinical trials years ago, drug by Eli Lilly's product. That was a drug that inhibits gamma secretase, something that is believed, like enzyme believed to be implicated in the development, accumulation of amyloid proteins in Alzheimer's patients. In preclinical studies, it seemed to have all the hallmarks of activity that you would expect for a drug that works at that mechanism, but yet when it was put into clinical trials, in fact, patients in the treatment group had earlier onset of dementia, and it also had elevated risk of skin cancer, something that wasn't expected from the preclinical research. So the net upshot is that oftentimes clinical trials are our first occasion to actually test some of those theories that are guiding our drug development. And as you might imagine, partly because of the nature of how difficult it is to understand disease processes, many of our theories turn out to be wrong, and we only learn that through the clinical trial. So that's one reason why it's so hard to translate drugs, is we don't really understand biological reality very well. But there are a couple other reasons too. Another has to do with the fact that you can think of trying to find a drug as kind of akin to looking for a needle in a haystack. Years ago, there was an airliner that went down in the Indian Ocean, Malaysian airliner. And I believe to this day, they still haven't actually located the airliner, but there was this huge sweep of the Indian Ocean to try to find debris to locate that downed airliner. And one of the sort of big challenges, you can sort of think of that process of trying to find that downed jetliner as akin to trying to find the rare combinatorial of a drug at the right dose, in the right indication, on the right schedule, in the right type of patients. And what the task of early phase drug development is about is scanning that vast landscape, that vast combinatorial landscape, in order to find a little bit of signal that you might have something promising, and then to follow up with all your heavy equipment, your phase 3 clinical trial, now to vindicate that promising signal. So if you, again, sort of working with this analogy, if you think about it, if you're trying to scan the Indian Ocean really, really quickly, you're not going to want to deploy really expensive, fancy equipment to any one spot until you have some signal of promise. And so you're going to want to use methods that are fast and frugal. What does that mean? Well, fast, you're going to use surrogate endpoints rather than clinical endpoints. And frugal, you're going to want to employ trials that use relatively small sample sizes. Well, these are precisely the kinds of conditions that create false positives. Add on top of that it's like a diagnostic test where you have a very, very low prevalence. As you know, if you have low prevalence, even with a very specific diagnostic tool, many of your positives will turn out to be false positives. And so that's another reason why a lot of drugs look promising early on, turn out not to actually be safe and effective when they're put into rigorous tests. And there's a third factor that I think it's important to bear in mind, and it has to do with random variation. So every time you run a clinical trial and you measure an effect, you're measuring a real effect, but you're only estimating it because there's random variation around that real effect. And so if you are testing a bunch of different drugs, some of those drugs are going to give a really, really large effect. Some of them are going to give a small effect, and you don't necessarily know in advance whether those large effects reflect random variation or a true effect. You'll only know that when you try to replicate that finding and you observe potentially regression back to a smaller effect, that's a small, that's actually not a real true effect of a drug in an early phase study. So if you're a rational researcher, you test 10 different drugs, one of them gives you a big signal, and the other 9 give you a small signal. A rational researcher will follow up on those positive trial interventions. And chances are, if it's due to random variation, that positive effect is going to regress back to a smaller effect. And so we see that all the time in clinical research, where big effects in early phase clinical trials partly are reflecting random variation. When we go back to retest those effects, we see a regression back to a smaller effect. So these are all statistical and scientific reasons why it's just so hard to develop a new drug. So that's tough enough. Now add to that, on top of it, that there are any number of different practices in the way that we do research that make that process of trying to discover a safe and effective drug just that much harder. And I find it often helpful to kind of think about research as having as a cyclical process that has sort of three different stages. Design, you design a clinical study, you report the clinical study, researchers now read that study, they absorb it into decisions about the design of a subsequent study, and the cycle begins anew. And there are any number of different factors that can lead to error in the process of trying to discover safe and effective drugs in clinical development. At each one of those steps, let's talk first about design. Anytime you run a clinical trial, what you're really trying to do is to isolate a cause and effect relationship between the treatment and disease response. And there are any number of different factors like bias or noise or artifacts, et cetera, that can interfere or confound with your ability to relate a drug to a particular treatment effect. And that's why when we design clinical trials, we often design them in ways that are intended to reduce those confounding effects or to address various threats to internal validity. So for example, we often use random allocation, treatment allocation, a priori power calculations, blinding. All of these are meant to protect the internal validity of the study and to prevent us from advancing a drug into further development on the basis of spurious, biased findings in early phase clinical trials. Well, unfortunately, a very large volume of the clinical trials that we run tend to be pretty small or tend to use substandard research methodologies. This is just, almost a random figure. It's from a paper that I published about various clinical trials of the pain drug pregabalin. All you have to see in this slide is that, we had a really, really large sample of trials, 238. I hope you can see my cursor there. But only about 47% of them were single center studies, and 70% of them had sample sizes of less than 100. So a very large fraction of the clinical research enterprise, it's not clear what fraction, but a large fraction of the enterprise uses methodologies or standards that are prone to giving us unreliable findings. Now, of course, the second issue is addressing threats to construct and external validity. We're not just trying to show an effect in a clinical trial. What we want is to show an effect in a trial that will generalize to a clinical practice setting. And there are any number of different factors that can frustrate our ability to generate a reliable and generalizable finding from a clinical trial that will generalize to clinical practice. Here's just a quick illustration from the realm of cancer. If you look at the age structure of a typical registration clinical trial that is a trial leading to approval of a cancer drug, you can see that about 39% of patients in those trials are over the age of 65, 70% are over, or 20% are over the age of 70, et cetera. If you actually look at the age structure of cancer patients, though, you can see that the registration trials tend to under-enroll patients who are older, patients that have an age structure that's more similar to the realm of cancer research. And that partly explains why some of our findings in registration trials fail to generalize into clinical practice. Sorry about the garbage pickup outside my window. Another big issue is the use of surrogate endpoints. In many realms, registration trials are based on surrogate endpoints, indirect measures of clinical benefit. In cancer, for example, only about a quarter of cancer drugs that are approved demonstrate a benefit on survival, a clinical endpoint within 3 years of regulatory approval. So there you have this kind of mismatch between the circumstances under which you test the drug and the kinds of outcomes that you're hoping to generalize to. And that explains part of the challenge with clinical development. Okay, on to reporting. There are any number of different challenges with reporting. One common issue is that there's incomplete reporting in clinical trials. A good example of this is a publication in 2009 where the researchers looked at a sample of clinical trials that were published in top-tier medical journals, and they looked at the concordance between the primary endpoints in the publication and the primary endpoints that were registered prospectively on clinicaltrials.gov. And what they found was that roughly a third of clinical trials showed evidence of having altered the primary endpoint between the registration record and the publication, thereby potentially threatening the statistical integrity of the study. Another really common challenge in clinical research is the issue of non-reporting, of actually not even publishing the results or disseminating the results at all. So for example, my team led a project years ago where we looked at the fraction of clinical trials, pre-licensed clinical trials, so trials of drugs that have not yet been approved. We wanted to look at the fraction of trials that went on to get published within 5 years of the completion of that clinical trial. And what we found was that for pre-licensed trials of drugs that go on to get approval, about 25% of those trials never actually get published, or at least they don't within 5 years. But the numbers are dramatically higher for non-publication. Only about 37% of pre-licensed trials of drugs that do not go on to get approval ultimately get published. So there's a lot of information that actually never gets disseminated. And again, if you get back to that Alzheimer's slide and the mechanism slide, where I talked about how these trials are your first test of the mechanism, the mechanistic hypothesis that's driving drug development, that non-publication really frustrates our ability to really learn more about the mechanism so that we can revise our theories and hopefully design more effective interventions for clinical development subsequently. Finally, there's the issue of uptake of research findings, sort of what I call the bottom of the iceberg. It's one thing to design and report a study properly. It's another thing to get people to actually read it and to interpret it properly and to apply it in their decision making. And there are a number of examples that illustrate some of the problems with uptake. This is a paper that was published by my friend and colleague, Dean Ferguson at the Ottawa Health Research Institute. Basically, what they looked at is all the placebo-controlled trials of a drug called aprotenin, which is used to control bleeding during surgery. So again, these are placebo-controlled trials, so patients are being allocated to a no treatment arm. And this is a cumulative meta-analysis. And basically, if you're to the left of this line, you're showing a clinical effect, a clinical benefit. You can see that there's a huge amount of redundancy in the research enterprise. By the time you get to, say, the 10th trial or the 12th trial, somewhere around here, it's pretty clear that you've demonstrated efficacy. It's hard to sort of understand what the justification or rationale is for running those subsequent studies and for allocating people to a placebo arm. That's a good illustration where research communities do not appear to have moved on in spite of the fact that there's convincing evidence that the drug is actually effective. My own team has also looked at this in the context of the relationship between exploratory phase 2 type trials and confirmatory phase 3 type trials. This is a publication we did on the drug pregabalin, which I mentioned earlier. These are all the different indications in the vertical axis that pregabalin was tested in. Each one of these nodes is a clinical trial. And the triangles are phase 3-like clinical trials. And the circles are phase 2-like clinical trials. So think of the circles as being hypothesis generating, and the triangles as being confirmatory clinical trials. Now, ideally, the way you would want things to work is you do a hypothesis generating trial, you get a positive signal, like for restless leg syndrome right here, and then you follow it up with a confirmatory clinical trial. That's good because there's a short period of time where there's uncertainty about whether or not pregabalin is useful for restless leg syndrome. But the reality is that because it's so hard to do those confirmatory studies and so easy and cheap to do those exploratory studies, and remember, I said that these exploratory studies tend to lead to false positives because they use fast and frugal methods. You have a lot of cases where you have these exploratory studies, like here for irritable bowel syndrome, that generate a signal of promise, but there's never any follow-up with a confirmatory clinical trial. In fact, in our publication, this was in JAMA Internal Medicine, I think 2017, we found that for 63% of the indications for which there was generation of signal in exploratory phase 2 type trials, for 63% of those cases, that clinical trials was not followed up with a proper confirmatory trial within phase 3, meaning that for many of these indications, alcohol dependence, et cetera, or low back pain, et cetera, the clinical community was left uncertain whether or not the drug was actually going to be useful, pregabalin was going to be useful for treating those indications or not. And nevertheless, oftentimes those exploratory trials, the results thereof are incorporated into clinical practice guidelines as recommendations. Here are 6 different recommendations or 7 in clinical practice guidelines that were made on the basis of exploratory studies that were not followed up with proper confirmatory trials. So these kinds of concerns motivated me with a bunch of collaborators, Deborah Zarin and Steve Goodman, to write a paper about the harms of uninformative clinical trials. And in this paper, we lay out 5 different conditions that any trial needs to fulfill in order to meet the standard of social value or informativeness that it should meet before you go forward and enroll patients. Number 1, you have to be addressing an important research question. You have to, number 2, know that your study is feasible, that you're going to be able to recruit enough patients. You have to be using a sound research methodology, one that is suited to the conclusions you want to draw. There has to be analytical integrity. And 5th, there should be a plan and a practice of full and prompt reporting of your outcomes. So I want to conclude this section by just saying that drug development, in addition to dealing with statistical challenges, also contends with a lot of research methodologies that are inefficient or that lead to or under-informative clinical trials. Let's move now to the center of this talk, pandemic research exceptionalism. So all this is to say that for years, my research group has been thinking about various ways that we, in a sense, underutilize the volunteerism of patients when we do clinical research by using designs or by not reporting trials properly, thereby failing to maximize the risk-benefit ratio for those patients. So having been sensitized to those issues, when the coronavirus pandemic began in January and February, as new cases were beginning to accumulate, particularly in the West, I was already kind of sensitized to the fact that there are any number of different deficiencies in the ways that we do research, even under non-wartime conditions, such that with the urgency of the pandemic, you would expect potentially there could be even amplification of these kinds of research inefficiencies and or deficiencies. Now, what was particularly concerning was not just that risk of substandard, methodologies being quickly put into the field, but also it was that a number of different commentators, some very prominent, often in top-tier medical journals, argued that because of the crisis conditions that we're facing, we really have to kind of make a compromise with the way we do research, that because it is so urgent to do this research, we really need to kind of relax some of the kinds of fastidious standards that we normally maintain when we do clinical research. And so together with my co-author, Alex London at Carnegie Mellon, we sort of wrote a paper about what we were calling pandemic research exceptionalism. Basically the idea that crisis conditions essentially license researchers, sponsors, host institutions, regulators, et cetera, they license a relaxation of the methodological standards. And my co-author and I thought that there were a number of different underpinnings for that research exceptionalism that we thought were problematic underpinnings. The first was this notion that, if you're facing crisis conditions, getting some information now, even if that information is flawed, is better than having no evidence at all. That's a totally understandable way of thinking. It was, to be frank, it was something that I was sort of tempted by initially to think, okay, maybe we should relax these standards so that we have at least a little bit of information. But that notion is probably a sound notion if in fact we were confident that the errors in our clinical trials were completely random errors as opposed to biased errors. That is, that the error, if we were confident that there wasn't any directionality to the errors in our clinical trial. But in fact, that generally isn't the case with clinical research. So again, you can go back to this question of whether a little bit of flawed evidence is better than no evidence at all. And although it seems very contrary to our intuitions as scientists, in a realm where research findings can be so easily put into clinical practice, actually a little bit of information can be worse than no information at all. So again, I just want to go back to this example of pregabalin, where you have numerous cases of interventions for which you had a little bit of information from probably biased small clinical trials suggesting pregabalin might be effective for any number of these disorders: acute anxiety, lower back pain, depression, et cetera. And on that basis, you have an uptick of off-label use of pregabalin and or uptake in clinical practice guidelines, even though we know that many of those findings are likely, at least half of them are likely to be overturned were those interventions put into more demanding, rigorous, confirmatory clinical trials. So a little bit of information can actually be harmful, all the more so when these are interventions that potentially entail side effects, when they are potentially costly, and when the cost of deployment is at the expense of having resources available, whether those are actually financial resources or personnel available to deploy other kinds of urgent interventions that you might have in a pandemic setting. Now, add to that that there's this issue that once you have a little bit of information out there that suggests the promise of an intervention, very quickly patients and or practitioners begin to form preferences about use of that treatment. And that may actually interfere with the willingness of patients to enroll in clinical trials where they might be randomized to a placebo arm. It might interfere with physicians being willing to recruit patients into clinical trials if they think that what's going to be best for their patients is to get access to the new treatment. Now, that's not always a problem so much with drugs that are pre-licensed clinical trials, because after all, you can't write out prescriptions for drugs before they're actually approved. But in the case of COVID-19, where many of the interventions that were being tested initially in the early days were off-the-shelf repurposed interventions that had already been approved, hydroxychloroquine being a good example. There, if you engender treatment preferences by generating a little bit of evidence suggestive of promise, you potentially frustrate the ability to actually accrue enough patients to those demanding, large confirmatory trials to quickly confirm or disconfirm the clinical promise of that intervention. So a second underpinning of that research exceptionalism, I think, my co-author and I thought, is a perception that often recurs in pandemics or in epidemics, that there's something unethical about randomizing patients to a placebo or to some other treatment, especially when you're dealing with a potentially lethal condition. So, certainly this was a discourse in the context of the Ebola outbreaks, various Ebola outbreaks, particularly in 2014, 2015, where many groups, including some of my colleagues, my bioethicist colleagues, argued against the use of placebo comparators in those Ebola trials, even though there wasn't an effective standard, standard of care available. And I think what many of these people who support the second underpinning forget is the notion that really the whole function of clinical equipoise is to minimize the tension between doing care and doing research. If your study is in clinical equipoise, what you are essentially doing is ensuring that no patient is going to enroll in this project of advancing our knowledge about the efficacy of new treatments while compromising on their accessing standard of care. No patient has to go below standard of care in order to advance our knowledge. Another sort of way of kind of capturing the intuition behind clinical equipoise is the idea that no intervention should ever be tested if you can foresee that that intervention is going to be superior to the standard of care. So again, let's go back to Ebola, or let's go back to various other infectious disease. There are any number of different cases where interventions have been tested in randomized controlled trials and not merely not shown to be more effective than placebo, but in some cases shown to be actually even worse than placebo. So in the realm of HIV vaccine development, there was a clinical trial, a large phase 3 clinical trial of an adenoviral-based vaccine candidate where there was higher rates of seroconversion in the treatment arm versus in the placebo. So the bottom line is it's erroneous to think that allocation to a treatment arm is giving patients an advantage over allocation to a placebo arm. In fact, if you go into areas that are particularly difficult to innovate in, if you go into areas like Alzheimer's disease or Parkinson's, where one drug after another have failed clinical development, at least our research from my group seems to suggest that you're actually better off probably being in a placebo arm. The best place to be in a clinical trial is in the placebo arm. So bottom line for the second underpinning that there's some sort of conflict between treatment and care, if you're confident that your randomization meets conditions of clinical equipoise, that really shouldn't be a concern. In fact, enrolling in a clinical trial ought to be an effective way of advancing your care, your pursuit of your own care, while also generating reliable information for informing future healthcare decisions. So the third underpinning of research exceptionalism, at least my co-author and I thought, is this notion that you can think of research as kind of this free market where all researchers are kind of free to exercise their discretion to pick whatever hypothesis they want to test and to pursue it. And under non-wartime conditions, non-pandemic settings, that kind of makes sense. It's good to give researchers a lot of discretion in terms of what research they pursue. But ultimately, we have to keep in mind that research, number one, is a public investment. It's not just that those researchers are directly funded by public funding bodies. They're also indirectly funded by public funding bodies. Pretty much anyone in Canada, also in the United States, who is a clinical investigator is the product of decades of government investment in their education and in their training. And so any kind of society is going to want to make sure that that scarce talent pool is allocated in a manner that is going to maximize the return on that public investment. And accordingly, researchers bear obligations to make decisions that are going to ultimately serve the public good. Ultimately, scientific research, it's not something, we all kind of have our own curiosity, and it's true that lots of researchers are driven by curiosity, but ultimately what really sort of sanctions our social investment in research is the notion that generating knowledge about new interventions is a public good, that there's social value. And accordingly, when we think about how to deploy researchers and what decisions individual researchers should make, that's going to be constrained by the expectation that, again, that's going to return some value for that investment. So with those sort of three different underpinnings dismantled, let's go through these different 5 standards that my co-authors and I articulate for a trial to meet the condition of informativeness, and look at some examples where we see a clear demonstration of what we call pandemic research exceptionalism. So let's take that principle, that criterion of importance. At the time that we wrote our article, which I think was in late April, there were about 15 clinical trials that were registered on clinicaltrials.gov that were registering hydroxychloroquine. In fact, just yesterday or day before yesterday, there was an article in Stat News anyone that you need this volume of clinical trials in order to demonstrate whether or not hydroxychloroquine is effective and or safe. So a lot of those trials are actually duplicative. They're not really answering questions that are important questions because there are other trials that are already addressing those questions. So that seems like a real misappropriation of social resources there. Let's go to the principle of, or the condition of sound research design. You can pretty easily find examples of clinical studies that did not deploy sound research designs, many of which ended up being published in top-tier medical journals like the New England Journal of Medicine. This article in particular really gets under my skin. This was a case series of compassionate use of the drug remdesivir. Siri thought, when I say remdesivir, Siri thinks that I'm calling her, so sorry about that noise. So what really got under my skin about this was that this wasn't even a study. It wasn't based on a clinical trial protocol. There was no randomization. There was no blinding. It was just a series of case reports. And yet New England Journal of Medicine, if you actually go to their submission guidelines, they state really clearly that they only publish research as opposed to a series of case observations. And so here's a good example where probably a small study like this that's not using sound design engenders treatment preferences, making it that much harder to potentially accrue patients to proper randomized controlled trials of the drug remdesivir. Other examples, there was a study that is underway right now, led by a team in Amsterdam, testing whether TB vaccine can confer some protection for SARS-CoV-2. In this study in particular, they're using an endpoint of absenteeism rather than seroconversion, merely because they can't afford to pay for the costs of testing that outcome. They're also not using a proper blind in this study, so research subjects are going to be unblinded. And I think what worries me about this is that it might be a really good hypothesis to test, but ultimately, neither a positive nor an inconclusive finding will really settle the question about whether or not this vaccine approach is going to be effective. So it's not clear to me that this is really moving the goalpost or moving the needle in terms of understanding how to manage COVID-19. Finally, that same paper or that same report by Matthew Herper in Stat News that I mentioned yesterday, about 39% of the clinical trials that are registered on clinicaltrials.gov that are testing COVID-19 treatments have enrollment sizes of less than 100. So a lot of them are small studies that are at risk of producing spuriously large effects due to random variation. Let's look at that third issue, analytical integrity. It's easy to find cases of trials in the COVID-19 context that violate this condition. This was a small study that got picked up in the New York Times and, in my opinion, uncritically reported by Denise Grady. Yeah, there was a very positive series of quotes from a commentator at Vanderbilt that this is going to send a ripple of excitement out through the treating community. If I were a garden variety doctor, seeing patients, I'd read a quote like this from someone, famous infectious disease expert, and I'd think, I'm going to give my patients hydroxychloroquine. So, within about literally a half hour of reading that New York Times report, I dug up the preprint, and the preprint linked to a registration record in a Chinese clinical trial registration. And I noticed pretty quickly that there were some pretty stark discontinuities between what the researchers said they were going to do and what they actually reported in the preprint. For example, the registration record said it was a 3-arm study. The preprint was 2-arm study. The registration record said there was going to be 300 patients in the trial. The preprint reported on 62 patients. The registration record said they were using a PCR endpoint. The preprint reported some sort of, in my opinion, bizarre symptom endpoint that was kind of unclear and opaque. And then finally, the registration record said that it was a double-blind study. The preprint, it was a little bit unclear whether or not it was double-blinded. The language was a little bit ambiguous there. So surely if it took me about 15 minutes or a half hour to find that, our experts at Vanderbilt and our reporter at the New York Times probably could have dug that up quickly to identify that. But the larger point here is that there are often, threats to analytical integrity in clinical trials in the COVID-19 context. Fourthly, there's the issue of complete and prompt reporting. At the time we wrote our article, there weren't really any cases yet because the trials were all getting started, but we knew they would accumulate, and there have been any number of cases where reports have been sort of sped to the public either through preprint servers or even worse, through press releases that are offering incomplete pictures of the evidence for safety and efficacy of new treatments. This happened, for example, with Moderna's press release around its COVID-19. They released results of 8 or 9 patients when the trial had enrolled about 150 patients. Their stock value went up dramatically. From what I understand from this news report, there were some prominent people at Moderna that sold their stock, that used the occasion to sell their stock. Also, you saw this in the case of remdesivir. Here, this is a study that is co-funded by the US National Institute of Allergy and Infectious Disease. That study was stopped early. When it was stopped early, there was a press release. It was not a bad press release, but it took probably about 3.5 or 4 weeks between that press release and the actual publication of the results, leaving the research community uncertain about how to evaluate whether remdesivir had shown efficacy and or safety. Finally, I want to talk about feasibility. Again, this was a theoretical issue at the time we wrote our paper. Now results are starting to come in, and you can see some examples of feasibility issues. Here's a case of a trial that NIAID was running of hydroxychloroquine. The registration record said that they were going to enroll 2,000 patients. That study got underway, I think, in early May. The study's actually already been halted. At the end of the day, they enrolled about 20 patients in that clinical trial. So, a good example of a trial where it's not clear there was a lot of feasibility, although in fairness to these researchers, they couldn't have anticipated that the results of the recovery trial would raise questions about hydroxychloroquine. On the other hand, why run a study that is going to be almost duplicative of a really well-designed study that's already underway? So the bottom line is, at least in my opinion, urgency is no justification for relaxing standards of trials. So that raises the question of what we ought to do under urgent conditions, because it is really, really hard to do clinical research under optimal conditions. And I'll get through this really, really quickly because I want to make sure we have time for questions. So I'll just say that many of the lessons we're learning in COVID-19 are lessons that have already been rehearsed in the context of other outbreaks like Ebola, where a lot of clinical trials ended up being basically uninformative because they didn't use proper control groups. But I want to offer a series of recommendations. Number one, sponsors and public funders should really seek to pool resources and prioritize those rigorous clinical trials like master protocols that are going to give us a really, really clear signal of treatment efficacy and safety. So the temptation is for funders to try to sort of disperse their funds to a lot of groups. More people are happy, there's less complaints from, parliamentarians in different ridings if the money is dispersed widely, but actually that's going to run counter to our scientific goals. What you really want to do is to concentrate your resources in a few studies that are going to be able to accrue a large enough sample size to test rigorously. And master protocols have a lot of advantages. Normally the way drug trials would work is you would be testing a drug against a placebo. If you're testing 4 different drugs, they're all being tested against a placebo. With a master protocol, basically you're folding all of those evaluations into one clinical trial. So number one, you're eliminating 3 different arms. So you've gone from 8-armed study, essentially, to a 5-armed study. So you're reducing the number of patients you need to enroll in that clinical trial by some degree. And on top of that, you now have comparative effectiveness information on each of the different drugs, because you're using the same randomization to assign patients to each of those different drugs. Whereas under these conditions, you really have to worry about confounds when you're trying to compare the effect size for drug B against drug A. So there are a number of really impressive master protocols that are underway. Recovery in the UK that many of us have heard about already. This was the trial that led to the demonstration of the efficacy of dexamethasone. And WHO has also launched the Solidarity trial. There are a number of other examples, and there's a Canadian arm of the Solidarity trial that I believe is enrolling as well. What can physicians and scientists do? Well, I think they ought to try to resist the temptation to write prescriptions off-label. They ought to try to affiliate themselves with large, well-designed clinical trials and to recruit and enroll for those clinical trials. And they ought to understand well-designed, well-evaluated clinical trials that are in clinical equipoise as a vehicle for delivering care while improving the knowledge base of our healthcare system. Academic medical centers can probably do a better job in terms of prioritizing the research that they host. They can be triaging trials and sort of cutting away those small studies that are going to compete for recruitment against those large, rigorous, well-designed clinical trials. And finally, regulatory bodies and public health bodies can have a role here as well. They can articulate standards for the design of studies, for outcomes that are used in studies. FDA has done that to some degree successfully. They can also resist the urge to approve drugs on weak evidence. FDA has done a very bad job of this. Health Canada has done much better, in my opinion. FDA has granted emergency use authorization in several cases where there was pretty flimsy evidence. And they can also, again, like academic medical centers, they can sort of triage and prioritize those clinical trials that are likely to have the biggest health impacts. That's within their remit, given that they have scarce resources. They ought to bump to the front of the lines those trials that are likely to move the needle. So in conclusion, the key to tackling the challenge of urgency is not to relax our scientific standards, but rather it's to constrain some of our discretion as scientists so that we are coordinating our activities and getting the most value per patient enrolled in clinical research. Now, I want to close with a quick coda. One of the most gratifying things about publishing this article was getting an email from an environmental scientist who told me that everything I said in that article was exactly consistent with the issues he encounters whenever there's an oil spill and environmental scientists rush to the oil spill to try to evaluate containment strategies and various other methods of protecting wildlife. A bunch of small, poorly designed, uncoordinated studies are launched, the oil spill ends, and people still aren't clear about what the most effective way to deploy an intervention. So the broad point that I want to make is, yes, this is all about pandemic research exceptionalism, but it's a broader point about the nature of how to do research under crisis conditions. Now, I just want to close by coming back to the ethical themes I started with. Our task is that we want to protect research subjects from undue incursion, from social objectives. That's part of what clinical equipoise is doing here. And secondly, we want to make sure that there is good integrity of the flow of information from the clinic to the people who are going to be using that information for making life and death decisions. And so ultimately, when we think about what the task is of research ethics, what our task is of research designers, really what we're talking about here is maximizing the efficiency of our research system. That is, how can we get the most amount of value in terms of public health impact with the fewest number of scientists and the fewest number of patients enrolled in that research enterprise. I'll stop there. Thank you.
Great. Thank you so much, Jonathan. So I'm going to open things up now for questions. So feel free to put your questions into the Q&A. And maybe I'll take the chair's prerogative and ask something. And I guess what I struggle with is, as you well pointed out, we seem to keep revisiting these same themes during different crises, whether they're an oil spill or whether they're Ebola or now with coronavirus. And what do we do to get buy-in so we're not redoing this over and over again? Do we need some sort of, do funders have to have sort of a framework or something where there's a little more plug and play? So rather than panic, you're having a more thoughtful approach to these crises?
Yeah, thanks. So yeah, it's a very important question. How can we be on top of this when we get a resurgence, which it may very well happen, and or for the next crisis? So in fairness, I mentioned earlier that, after the Ebola outbreak, many of these lessons were learned, and they were learned by the WHO. So after that outbreak, the WHO actually prepared a master protocol, sort of like I don't know whether people here have ever done the Mad Libs, it's like a short paragraph with a bunch of, empty slots that you fill in, sort of a game that you play with kids. That's what their protocol was like. It was like a Mad Libs for evaluating an intervention in an outbreak, and you fill in the drug and you fill in the outcomes. And of course you have to adjust the statistics according to the dynamics of the outbreak. But I think that any research system ought to be thinking about how to create these master protocols in advance of outbreaks so that they can be quickly pulled from the shelf and deployed when there's an outbreak. That's number one. I think research preparedness is also really key. I think there's a tendency to think of research as this kind of discretionary thing that, we fund because it's kind of nice to have, it's sort of like, I don't want to denigrate art. Art is an important part of my life. My wife is a musician. But I think that, it's important to recognize that science is not just a kind of a side activity for a healthcare system. It's an integral part of our healthcare system. As I said earlier, we want to make sure that our healthcare dollars are being deployed properly. And so accordingly, it's really important not just to have a pandemic strategy, a preparedness strategy for deploying treatments or for deploying social health, public health measures. It's important that there be a research strategy that's planned. So researchers, are sort of ready to be deployed in the event of an outbreak. You're not trying to create those networks anew or de novo when a new outbreak occurs. Sort of like the reserve army. So I think that's a second thing. Regulators obviously have some role to play here in pandemic preparedness. And there was another point that I was going to make that somehow slipped my mind, but I've probably gone on too long about this anyway, so maybe it'll come later.
Okay, so another question that's come up is, what suggestions do you have about research waste that results from non-publication of study results?
Well, there are a lot of different suggestions. I mean, I think that number one, in my opinion, there have been many people who have argued, and I agree, that non-publication of results of trials that have actually completed is a violation of scientific integrity. It ought to be considered a form of scientific misconduct. That's different if you start a study and you only enroll, 5% of your sample population, so you didn't actually really test the hypothesis. That's, or something goes wrong technically. So there can be exceptions, but generally, I think if we really understood non-publication of results as a threat to scientific integrity and a form of misconduct, it might change things. I think if researchers understood that properly, if there was some enforcement mechanism, it would change things. I also want to make clear that there are a number of different vehicles by which you can, report your results. So, it can be a pain to try to publish inconclusive or ambiguous findings in medical journals. All of us researchers, we're busy. We have different things we're trying to do, but nothing is stopping us from depositing those results in clinical trial registries like clinicaltrials.gov. So there are a number of different ways that results can be deposited besides going to the effort of publication. And finally, I think academic medical centers ought to be monitoring what fraction of the trials that they host are getting published, or at least having the results deposited within 5 years. Ben Goldacre's group in the UK has tried to kind of monitor that and create report cards for academic medical centers. Some do better than others, and I think that academic medical centers should be competing with each other to have the highest rate, or really trying to sort of compete not to be at the bottom of that list.
Great. Well, I am very conscious of time, and I know people are very busy. So maybe I'll just ask one last question before we close, and that is, are there any limitations or exceptions that are acceptable during a time of crises when you're doing research?
I'm not sure if I completely understand the question.
Well, no, are there any corners that you're allowed to cut during a crisis because of the nature of the crisis? Or is the short answer no?
Well, in ethics, almost never say never, right? It's like a paradox. You can't even say never then. So the answer, of course, there's got to be some cases where, where you might have a moral justification, for doing things differently than you would under non-urgent conditions. But I think what's really, really key to understand is that, if you're doing research, that research, in order to sort of deserve the name of science, has to have certain properties, right? There has to be transparency about what you're doing. You have to be measuring things that accord with what you're trying to measure. Otherwise, you're not doing science, you're doing something else, right? And so, to some extent, I mean, there may be interpretation of, how you trade off feasibility with, gold standard scientific methods, but you ought to be using scientific methods that are concordant with your objectives. And I think, I just want to come back to this idea that, often the conditions where we're tempted to relax our standards, if we thought outside the box and said, hey, maybe it's not about me just running my small clinical trial, but my joining with a team, it might turn out that we don't need to relax those standards. We just have to think differently about the way we orchestrate and organize our work.
Right. Great. Well, unfortunately, I'm going to have to cut things off there, but I want to thank Jonathan very much for speaking with us today and for this excellent talk. I personally really enjoyed it. I just want to remind people who are listening that there will be a survey sent out. So if you could respond, that would be great. Want to get your feedback and that what other types of topics you'd be interested in hearing about. And up on the screen right now, there's some contact information if you're interested in more information about N2. So I'll leave it there, and thank you once again, Jonathan, for a really informative hour. Thank you very much.